Let us talk about the types of meiosis and its significance. So, let us take types first. First is known as gametic meiosis. In gametic meiosis, diploid germinal cells, diploid, that means 2n germinal cells. Germinal cells are the ones which are capable of undergoing meiosis. These germinal cells undergo meiosis to form haploid gametes. So, this meiosis is taking place in diploid cells and the resultant cells are haploid and they are called gametes. This is required in case of sexual reproduction. So, this type of meiosis is seen in almost all the higher animals except protozoans. All animals show this type of meiosis. The second is known as zygotic meiosis. In zygotic meiosis, the individual or the organism is haploid. That means they have half the number of chromosomes or one set of chromosomes. They lead their life as haploid cells. When they have to reproduce, two such haploid cells would start behaving like gametes. They would fuse to form a diploid zygote. And the zygote undergoes meiosis. Again, it will be meiosis 1 and 2 to give 4 haploid cells which will start living as individuals. So, here meiosis is taking place in a zygote, whereas in gametic, meiosis is taking place in germinal cells to form gametes. The gametes here in this case of gametic meiosis fuse to form a zygote, and this zygote is diploid. It divides to form only diploid cells. So, the difference is here half cells, that is gametes, are formed to make a diploid zygote. Here haploid individuals fuse to form a diploid zygote and zygote undergoes meiosis. The third type is known as sporogenetic. Sporogenetic meiosis. It is seen in higher plants. Okay, well, let us write the examples of all. This is seen in animals, that is gametic meiosis in animals except protozoans. Zygotic meiosis takes place in lower organisms like algae, some protists and some fungi. Sporogenetic meiosis is in higher plants. This is very much similar to gametic meiosis. So, the organism is diploid. The cells of reproductive structures, they undergo meiosis. The germinal cells, they undergo meiosis. Germinal cells are also diploid. So, when they undergo meiosis, they form haploid spores or gametes. These gametes, they fuse. To to form a diploid zygote, and this zygote divides by mitotic division. Mitotic division to form the organism. This is seen in case of higher plants. So there are three types of meiosis. Significance, of this type of division, that is reductional division, it is important for sexual reproduction. So meiosis is responsible for gamete formation. And these gametes are haploid structures or half cells. Why this is important is that every species has a species specific chromosome number. So when these gametes fuse, fusion of gametes again gives that species specific chromosome number. Species specific chromosome number. If this number is not reduced, after fusion we will get some other kind of number. For example, in human beings, diploid cells have 46 chromosomes. These cells with 46 chromosomes, when they undergo meiosis, gametes are formed with 23 chromosomes, half the number. So egg has 23, sperm has 23. When these fuse, again that species specific 46 number is achieved. So because of this, there is maintenance of this species specific chromosome number. When we were talking of the process, we talked of crossing over. What was happening in crossing over? The genetic material of two chromosomes was getting exchanged. This results into genetic variation. So one very important significant point of meiosis is it produces genetic variations, and variations are important for adaptations. So these variations help in adaptations. So there are these four major things for which this process of reductional division, that is meiosis, is very, very important, mainly for sexual reproduction and genetic variation. Now let us compare mitosis and meiosis. We have done both the processes. Let us see in which categories or which important points they differ. So comparison of mitosis and meiosis. So let us take the points of comparison between mitosis and meiosis. We will make 3 columns to understand the point on which we are comparing, and then see what happens in mitosis and meiosis. So these are the points on which we will be discussing. Then we will see what happens in mitosis and meiosis. The first point is, in which cells do these divisions take place? So type of cell which undergo mitosis and meiosis. Mitosis takes place in all cells, whether those are somatic cells or germinal cells. In germinal cells also, in multiplication phase, only mitotic divisions take place. So we can write all types of cells. Whereas meiosis takes place only in germinal cells. Germinal cells are found in the lining of sex organs. Ovaries have those cells which we call follicles, and even testis, or in plants also there is germinal epithelium. The cells of this epithelium only can undergo meiosis. As a result of these divisions, how many cells are formed? So, number of cells formed. As a result of mitosis, 2 daughter cells, and in case of meiosis, 4 daughter cells. Which type of cells are formed? Type of cells formed. The cells which are formed, they are identical to the parent cells. So, identical to parent cells, and here they have half the number. So, they are different from parent cell. What happens to the chromosome numbers? Chromosome number. In case of mitosis, the daughter cells have the same number. So, same number as in parent cell. Whereas in case of meiosis, half the number as compared to the parent cell. Next point. Now let us come to the significant things. Let us take stage by stage. How many phases are here? Karyokinesis and cytokinesis, same in both, so we don't have to write about that. Here we have how many phases of karyokinesis? Prophase, metaphase, anaphase, telophase. Same here, prophase, anaphase. But here this division is taking place in one go. Whereas in case of meiosis, we have meiosis 1 and 2. So, the point of difference is phases. Here only one, which comprises of karyokinesis and cytokinesis. In case of meiosis, there are 2 phases, or you can write 2 stages or 2 parts. That is meiosis 1 and meiosis 2, each having karyokinesis and cytokinesis. So, it is double or 2 times. Let us talk about prophase. In case of mitosis, there is only one prophase. Here there are 2 prophases, prophase 1 and prophase 2. But the significant difference is, in case of prophase of mitosis, there is no crossing over. In case of prophase 1, there is crossing over. So, here we will write no crossing over, and this includes everything, that is chiasma formation, crossing over, exchange of genetic material, terminalization, everything is a part of this. Here crossing over takes place. When does it take place? Only in prophase 1, not in prophase 2. So, prophase 2 is similar to mitosis, but in case of prophase 1, there is crossing over. We have seen the 5 subphases. In prophase here, do we have subphases? So, we can write no subphases. These subphases can be written as early prophase, mid prophase and late prophase, but those are not the actual things, these are just the sequential changes. But here in prophase 1, we see 5 subphases. And in prophase 2, there are no subphases. So, only prophase 1 has 5 subphases. In prophase, because of this crossing over, there is variation in the genetic material also. Let us take metaphase. What happens in metaphase of mitosis? Spindle fibers are formed, chromosomes are arranged on the equatorial plane. Here also the same thing. So, no significant difference. So, let us take anaphase now. What happens in anaphase? The chromosomes get separated. Which type of chromosomes are getting separated here? The original and the copies. That means centromere is undergoing splitting. So, here chromosomes separate due to splitting of centromere. Splitting of centromere. This is important. So, let us underline this. Splitting of centromere is very, very significant thing here. In case of meiosis, meiosis 1, that is anaphase 1 if we talk of. What is happening in anaphase 1? The two chromosomes are separating. Which are these two chromosomes? They are homologous pairs. So, homologous pairs, they are not joined by their centromere. So, in anaphase 1, there is no splitting. Whereas, in anaphase 2, which is similar to mitosis, there is splitting of centromere. So, again important, no splitting in anaphase 1, but splitting of centromere takes place in anaphase 2. Telophase, the last stage of nuclear division. What happens in telophase of mitosis? Nuclear membrane reappears, nucleolus reappears, chromosome again becomes thin into chromatin fibers. Here also same thing. In case of telophase of mitosis, the cytoplasmic constriction also begins. Here also it takes place. Then let us take the next stage, that is cytokinesis. Cytokinesis, in case of mitosis, cytoplasmic division is only once. So, cytokinesis is only once, and here twice, 2 times. Cytokinesis 1 and 2. One more important thing, when are the spindle fibers formed? Spindle fibers are formed during prophase. Is there any difference in the orientation of spindle fibers? In case of meiosis, prophase 1 and prophase 2, the spindle fibers are right angles to each other. If first one is vertical, the second one is going to be horizontal. If first is horizontal, the second one is going to be vertical. So, that will be the difference in the orientation. 4 cells we have already written. So, these are the differences between mitotic divisions and meiotic divisions. Now, certain important terms which are in general discussed whenever we talk about cell division. One very important term is called HeLa cells. HeLa cells. These are significant for a reason that these cells were taken from the cervix of a female who was suffering with cervical cancer. So, the name of this lady was Henrietta Lack. She was suffering with cervical cancer, cancer of cervix. These cells were taken out for biopsy and studying it. Cancerous cells have the capability of dividing even outside the body. So these cells, the female died after some time when the cells were taken out for the examination purposes. The female died after some time. The cells were still kept in the laboratory and scientists kept working on those cells because the cells were multiplying in the culture medium. So that is why these HeLa cells are important. Second important thing that we talk about divisions whenever mitosis, meiosis are discussed, one is cancer. Cancer is defined as an uncontrolled growth of cells. How would the cells grow? They are dividing basically. So it is an uncontrolled cell division that results into cancerous growth, which we call tumor. These cells which turn cancerous, there are various theories, we are not going into the details of the theories, but when they turn cancerous, they lose the power of contact inhibition. Normally the cells, every cell is programmed. It has to divide by a fixed number. Every cell has it in, that thing in its genetic material. Say a cell has to divide 10 times. So every time it will go through cell cycle, interphase M, again cell cycle, 10 times. After that, it will come out and go into G0. But these cells, they lose this power and they remain in the cell cycle forever. That means they will go on dividing and that results into formation of this tumor. So cancer is due to cell division. Which type of division is taking place? The division is mitotic. All divisions which result into cancerous growth are mitotic division. That means all new cells which are formed, they are all same type, that is of the parent cell. Chromosomes are thickest in metaphase thickest in anaphase and shortest in metaphase. So when they are shortest and thickest, this is the best time to study them. Normally they are studied during metaphase. So cell division is arrested at metaphase and then the study of chromosomes would take place. How would we arrest the cell division at metaphase? One is colchicine treatment. So arresting can be done by colchicine. It acts as mitotic poison. It does not let spindle formation take place. So spindles to which these chromosomes are going to get attached and then separated will not take place. So chromosomes become short and thick, but because they do not get attached to spindle fibers, as spindles are not formed, they remain as it is and you can study them at this metaphase stage. Colchicine and other mitotic poisons we have already talked of when we were discussing mitosis. So some important things whenever there is a reference of cell division, that is the time when we talk of HeLa cell cancers and the stages when it is thickest and shortest. So we are done with all three types of cell divisions, that is amitosis, mitosis and meiosis. We have also seen the comparison between mitosis and meiosis, all diagrams, all important events, just revise them so that everything becomes clear. I hope this is enough to understand the complete cell cycle and cell division process.